Hello everyone, I am Dr. Bandyopadhyay, Assistant Professor of Biology. Today we will discuss about the techniques in biophysics. This part or this chapter is important only for the BS EEE 301 courses, which is actually for the EEE department third semester students. The paper is Biology for Engineers. So biophysics, bio means biological, physics means you all know what is physics. So biophysics actually is a combination subjects of biology and physics. So biophysics employs different advanced techniques to explore mainly the structure, the dynamic, the interactions of biological molecules. So how the biological molecules, like there are four different main biomolecules, which is the protein or the carbohydrate or the fatty acid or glycerol, the lipids or the nucleic acid. So how the biomolecules interact with each other. What is the structure? If you want to see the protein structure, we already have read that there is a different types of structure called primary, secondary, tertiary and quaternary. So if you want to observe the structure of the protein, then you need the biophysical techniques. If you want to observe particularly any microbes, you need any kinds of microbes, the bacteria, the fungi, the algae, the protozoa. You need microscopes for observation. To observe our own cells, the cells of plants, we all need the microscopes. So these are the part of biology. To actually analyze the concentration or volume of a fluid in the biological cells, to actually calculate the concentration of the DNA, isolated DNA from any living organisms, we need also some biophysical techniques. And another is a very new modern techniques which is called single molecule methods, like atomic force microscopy and other optical methods, which enables the study of forces and motion within an individual single molecules. The power of the spectroscopy in biophysics. So for spectroscopy analysis, at first we will observe that the spectrophotometry machine. This is a normal spectrophotometry machine. There are different types of spectrophotometer. So this machine is known as spectrophotometer, and we can break down the parts of the spectrophotometer word to help what the word means. Means spectro comes from the radiant energy or light energy. You need light. And the next part, photo, comes from light. So spectro comes from the energy, radiant or light energy. Photo means light, and finally meter refers to an instrument which is measured. You can see the detection is based on the light intensity, based on the wavelength, and you can generate the absorbed spectrum. This is used to in structure prediction as well as to know the concentration. This is the machine. So the spectroscopy techniques actually used the interaction between the principle is the interaction between the light and matter to unlock the molecule secrets. This is the molecule structure or concentration. By measuring how the molecules absorb the light or emit or scatter the electromagnetic radiation, the researchers can determine the structure, the monitoring dynamic variation or changes, and observe their interaction. So there are different types of spectroscopy techniques. One is UV-Vis spectroscopy. UV means UV light, ultraviolet, and Vis means visible. So in this there is a UV range in the range you all know, and another one is a visible range. So this spectroscopy actually analyzes the electronic transition in the molecules, revealing the chromophore environments as well as the concentration measurement. It actually follows the Beer-Lambert's law. And in IR spectroscopy, where probes or small molecules are used, small molecules used for vibration to determine the protein secondary or tertiary structure. Also nowadays it is used for 3D structure prediction. And then fluorescent spectroscopy. Fluorescent, it emits a fluorescent light, fluorescent color, fluorescent light, and tracks the molecular dynamics with exceptional sensitivity, monitoring protein folding, nucleic acid hybridization means 2 DNA where it binds, and real-time cellular processes. And currently nowadays it is very much used, which is the NMR or nuclear magnetic resonance. NMR spectroscopy actually stands the standard techniques for determination of atomic resolution structure of proteins, nucleic acids, small molecules in a solution. So the responsibility, the key main functions is complete the 3D structure determination without forming the crystallization. There are another technique for structure prediction which is the X-ray crystallization. But in this case, the crystallization is not required. Detection of the protein ligand. The ligand are specific molecules which bind a particular receptor or a protein. Binding and their interaction. The metabolic changes. There are metabolism. We already heard about the pathways, the glycolysis, Krebs cycle. So metabolic changes, photosynthesis also. Real-time monitoring of enzyme. We all know that enzyme is the biological catalyst which enhances the rate of the reaction. So monitoring the enzyme's reaction also performed by NMR. Microscopy. We all may be used in microscopy in our school also for visualization of any specimen. So there microscopy actually modern there are different types of microscopy. Scientist Joseph Frank got Nobel Prize for the discovering of cryopreserved microscopy. In the mind is a very less temperature to observe the cells. So there are some important microscopies like confocal fluorescent microscopy, which also eliminates the out of focus light to create sharp optical sections for better observation, enabling the 3D reconstruction. Super resolution techniques like STORM and PALM, which are actually achieve resolution below 20 nanometers, revealing the molecular scale organization of the cellular components. Atomic force microscopy uses the nanoscale probes. Probes means some small molecules which binds to map the molecular surface with sub-nanometer precision while measuring the forces between molecules, providing the insight into the protein mechanics or DNA topology. Next, holotomographic microscopy is actually used for construction of the 3D refractive index distribution without any leveling. You can see this is a microscope and visualization like that. So physiological techniques. There are some physiological techniques also known as electrophysiological techniques. You already heard about ECG or in heart, EEG in brain. So these are all the electrophysiological techniques which are actually used in the diagnostic laboratory. Electrophysiology is a study of the electrical activity in the cells and in the biological cells or tissue, which helps the scientists to understand how the signals are generated and transmitted. For example, the patch pump techniques, which are actually commonly used to record the ion channel in neuron. This techniques actually in cardiovascular, like in ECG, EEG, it is used in all. And then single molecule methods. Single molecule methods, you can see it requires also microscope. Actually allows the scientist to observe and to quantify or measure the behavior of a particular single molecules. It can be a single protein or it can be a single particular lipid molecules. So fluorescent correlation spectroscopy actually completes the imaging techniques of a particular with the help of fluorescent probe for a particular single molecules detection. There is also FRET, also single molecule fluorescent techniques, the total internal reflex fluorescent microscopy and confocal detection, which tracks the individual level particular single biomolecules. Then molecular modeling and stimulation. It is actually required a computational, means helps of a computer. Biophysics. This molecular modeling actually utilizes the computer stimulation, means computer programming and software, to visualize and predict the structure. You can see there is a structure of a molecule and interactions and behavior of the any biological molecules. It requires for that structure prediction like any organisms like evolutionary algorithms like AlphaFold and cleavage actually required for the to understand the evolutionary mechanism of a molecules. Mean how a particular molecules comes from the previous ancestral gene, and molecular dynamics which stimulates a molecular dynamics stimulation calculate the atomic motions by solving the Newton's equation for every atom, revealing how any biomolecules fold, bind the particular another molecule known as ligand or undergo any conformational changes over the nanosecond to milliseconds. Now the technology now it is actually very much improved. So it is integrated biophysics greater than the sum of all the parts. It's a multi-modeling. We already observed that for the multi-modeling imaging, combining the confocal microscopy and spectroscopy for structure detection, the structure integration using cryo-electron microscopy, I already told that, and the biophysics or enema dynamics measuring. It's also microscopy and spectroscopy combined, and live cell analysis, any integrated biosensor. Any particular single molecular tracking reveals the real-time cause and effects relationship between the molecular events or the cellular phenotypes, what we observe. The future of biophysical techniques, the high resolution, nowadays next generation super resolution matters, the image quality will be better, the AI integration, means the machine learning will transform the data analysis or predict modeling, and also faster acquisition and enhance their accuracy. Means if the accuracy rate will be enhanced if the quantum sensing or improved force spectroscopy will measure the molecular interaction with the single molecule precision under the physiological conditions.